It's beautiful, isn't it? There are few sights more majestic and awe-inspiring than that of a sea turtle gliding along the open ocean. Unfortunately, not only are sea turtle sightings becoming less frequent, but encountering one this healthy is also becoming less likely. In addition to the plethora of man-made threats they are facing, for the past 60 years, sea turtles have also been battling a deadly disease. With 5 of the 7 species of sea turtles already on the endangered list due to bycatch, hunting, and climate change, this might be the final stressor that seals their fate. Fibropapillomatosis, or FP for short, is a neoplastic disease that presents as benign tumors in a turtle's soft tissue. These tumors, which can grow as large as 30 centimeters, commonly form on the cornea, mouth, flipper, throat, and genitals. While the tumors themselves are not fatal, their presence can impede feeding, predator evasion, and reproduction. Often, as these tumors grow larger, the turtles lose function and die from starvation. The cause of FP has yet to be confirmed. However, it has been linked to a herpes virus, CHHV5, in conjunction with environmental factors. While it has been proven that the virus is present in asymptomatic turtles and can be traced back several centuries, the prevailing theory is that the industrial era has catalyzed its presence. Because FP cases are significantly more frequent near shores of developed areas, it has been stipulated that the excess nitrogen that runs off from industrial and agricultural waste accumulates in the ocean. The nitrogen is then absorbed by macroalgae, where it is stored as arginine, a known tumor-promoting chemical. This algae constitutes 90% of the turtle's diet. The turtle then accumulates large amounts of arginine. Once in the turtle's bloodstream, the arginine forms a vital part of the glycoprotein envelope. This envelope allows the virus to enter the host nuclei by circumventing checkpoints that would otherwise block the virus. Because only low levels of the virus are found in fully developed tumors, the disease is thought to be neoplastic, where it alters the genetics of the cell, thereby altering the cell signaling transduction network, allowing tumors to propagate without the virus. This remains mostly theoretical because the virus cannot be cultured in the lab. A cure remains elusive because the virus's genome has yet to be sequenced fully. A portion of the unsequenced genome contains the genetic code for its latency period, and without addressing this phase, any vaccine created would be ineffective. Currently, surgical excision is the only treatment option available. However, traditional surgery presented with a host of complications, and other techniques are currently being explored and implemented in more specialized veterinary clinics. Carbon dioxide laser surgery is now the surgical standard for FP treatment, as it has been deemed safer than traditional surgery. Layers of skin are removed using high-intensity laser beams. This method is preferred because it is faster, allows for hemostasis, requires no sutures, diminishes swelling, reduces the risk of infection, minimizes scarring, and has a better prognosis than traditional surgery. Also, the recovery time is often as little as 6 weeks. However, there are still limitations. Only 20% of patients are eligible for release. This is because multiple surgeries are often needed to remove all the significant tumors. Also, tumors tend to recur. Additionally, not all turtles are eligible for surgery. If their fibropapillomas are listed as a category 3, they are deemed too unstable for surgery and are often euthanized. Also, turtles must have a packed cell volume of at least 20% in order to undergo surgery, and often they come in with a PCV as low as 9% and must be rehabbed for at least a month. Lastly, the procedure requires full anesthesia, which is both dangerous and expensive. With all these limitations in mind, veterinarians have turned to human cancer therapeutics for an alternative and have discovered that because the disease is neoplastic and shares similarities with basal cell carcinoma, electrochemotherapy, or ECT, was a good treatment option. This technique is already being used on several other species. ECT uses a series of short, high-voltage electric pulses that leads to increased membrane permeability and makes it easier for the chemotherapy medicines to enter into the targeted tumor cells. Bleomycin is the drug most commonly used. The entire process can be completed with 2 sessions, with a 30-day interval in between, and complete regression has been observed 10 months after the second treatment, with no signs of recurrence after 1 year of rehabilitation. This method is beneficial because it is less invasive, less toxic, and only requires low doses of the medication. It circumvents invasive surgery entirely and instead relies on intralesional injections that target the tumor cells from within while protecting nearby tissue from damage. The procedure is relatively painless and requires only local anesthesia, making it less stressful on the turtle. ECT also has the additional benefit of triggering extensive tumor cell apoptosis and releasing tumor antigens that impede tumor recurrence. This treatment method remains in its trial phase and more studies need to be conducted as it cannot yet be used to treat multiple or coalescing lesions, and there are no protocols in place for intravenous use. The most recent treatment option being explored is photodynamic therapy. This has been used in humans to treat non-melanoma skin cancer and skin lesions caused by viral infections. This procedure involves a locally administered non-toxic photosensitizer, which is selectively retained in tumor tissue. In the case of FP, the photosynthesizer used is methylene blue. This uptake takes about 2 minutes, at which time the targeted area needs to be kept out of direct light. Next, the targeted area is irradiated with visible or near-infrared low-intensity light in order to activate the PS molecules. This leads to severe cellular damage, and within 4 weeks, the tumor becomes necrotic and either detaches or can be removed easily with tweezers, with no signs of recurrence. The treatment is administered in 2 sessions with a 15-day interval. This procedure has the benefit of being extremely low cost, ambulatory as there is no anesthetic, and little restraint is needed. It is also the most minimally invasive option available. Additionally, studies have shown that only the treated tumor is affected, meaning this treatment option is not toxic to the turtle as the chemicals do not spread throughout the body. Unfortunately, this also means systemic anti-tumor immunity is not triggered and each tumor must be treated individually. There are 4 key elements that need to be addressed if we are to alleviate the suffering of these turtles. Firstly, the high levels of arginine need to be reduced. One of the primary reasons arginine uptake has been increased is due to a non-native algae that has been invading the turtle's territory, most notably the Atlantic coast, Hawaii, Australia, and Brazil. Turtles need to eat twice as much of this algae in order to get the same caloric intake as the native algae being displaced. Researchers from the University of Hawaii may have found a solution. They have used a combination of a super sucker vacuum and introducing juvenile sea urchins and have reduced the presence of the invasive algae by 85% in just 2 years. Can this be effectively used on a larger scale? Will it be enough? Only time will tell. The second part of the puzzle is the virus itself. In order to create a vaccine, we must first understand how the virus works. This means we must be able to observe the virus in a laboratory setting, understand its latency period, and amplify the body's natural immunity towards the virus. Dr. Thierry Work from Honolulu may have just solved the first problem. In 2017, he and a team of researchers were able to grow reptile skin cells in the lab for the first time. This opens up new opportunities for studying the virus. Previously, the virus could not be studied because it cannot be cultured in the lab, and due to their endangered status, turtles could not be used in experiments. This offers a solution for both issues, and for the first time, scientists are able to observe and manipulate the virus. Currently, the short and long repeat sections of the virus's genome have not been sequenced, and until they are, we will not be able to fully understand the latent or dormant phase of the virus. Without knowing this, vaccines will not be able to properly target the virus during its latent phase. Because polymerase chain reactions, or PCRs, are not working, researchers at the University of Florida are now trying hybridization probes. The last component of this puzzle piece is the turtle's natural immunity to the virus. researchers have observed natural regression of tumors in turtles with category 1 fibropapillomas. They theorize that the turtles have a natural immunity to the virus that is only now being bypassed by the extremely high levels of arginine. If researchers can discover how these turtles are fighting off the virus, they may be able to develop a drug that can boost the turtle's immune system. Here is where epigenetics comes into play. Your epigenes are the switch that determine whether a gene is expressed or not, and can be influenced by your chemical environment. So, is the arginine suppressing the expression of this gene? Are there other pollutants that are responsible? Do all or most sea turtles carry the gene, or is it recessive? Is it more prevalent in some species? This last question is most likely true, as green sea turtles are 22% more likely to catch the disease than any other sea turtle species. A more important question is, can environments such as the Caribbean be selective towards this gene, since our nitrogen level increase is only moderate when compared to those of more developed areas, such as the US and Australia? In theory, because the stressor, the nitrogen, is being added to a lesser degree and over a longer period of time, this may be the conditions needed for turtles to adapt and overcome this disease. This is why it is important that areas like the Caribbean are also included in future studies. The third puzzle piece are the tumors themselves. Future vaccines may kill the virus, but because the disease is neoplastic, established tumors can propagate without the virus present. The solution to this lies in the systemic application of treatments like ECT and PDT, which can target the tumor without damaging healthy cells. The only limitation is figuring out the right dosage for intravenous use and studying the side effects of these procedures. The last puzzle piece is the simplest concept, but also possibly the most difficult to achieve. If we implement and properly enforce legislation that significantly reduces nitrogen runoff, it could alleviate the problem at the source. Another option that is somewhat controversial is using plants that have been genetically modified to draw nitrogen from the air. These plants would negate the need for excessive fertilizer use and therefore reduce the large amount of nitrogen in the water. Sea turtles are keystone species for marine environments all over the globe. They help to maintain the health of the ocean and seagrass beds, provide habitat, balance food webs, are a vital part of coral reefs' fragile ecosystem, and transport nutrients from the ocean to beaches. Green sea turtles are the only herbivorous sea turtles. They feed on seagrass and keep the seagrass beds healthy by increasing their productivity and the nutrient content in the seagrass blades. For the first time since the emergence of this disease, we have a clear direction to ending it for good. I have hope that ours is a generation that gets the sea turtle off the endangered species list.